Good day. Today we'll be discussing the African swine fever virus. Firstly, I'll be talking about possible samples that can be taken. Since the causative virus attacks the cells of its victims and induces a hemorrhagic fever, blood samples can be taken from the infected pig. Additional tissue samples can be taken from the lymph nodes, the spleen, and the tonsils. The samples can be taken up to 5 days after the fever has broken out. All the samples are sent to the laboratory where the virus is identified using various techniques like PCR.
Preservation and transport. Tissue samples to be put into formalin solution. Blood samples put into heparin. This keeps the blood from coagulating. With blood samples, serum should be collected using a sterile needle. Samples should be centrifuged or put in an upright position. Samples to be kept at cool temperatures. They are transported on ice or frozen gel packs. Freezing of samples is not recommended.
Lab tests that can confirm the viral infection. Serological tools are used to control and manage African swine fever. Since there is no vaccine against the infection, the presence of antigen induced by the virus indicates the infection. Serological diagnosis must be used in parallel with antigen detection techniques, which are PCR, ELISA, and IPT. The virus can grow in various cultures, which are pig bone marrow, pig leukocyte, and buffy coat cultures.
Septicemia is defined as an infection of the blood, so technically means it is an infection throughout the whole body. One must determine whether or not it is bacterial septicemia and not viral septicemia, as they both present with similar clinical signs. In order to deduce that it is in fact bacterial septicemia, you would take blood sample of the infected animal and grow a blood culture from it. If pathogenic bacteria is detected in the blood culture, it can be said that it is a bacterial septicemia. The morphology of the African swine fever virus is a large enveloped icosahedral virus, meaning it has 20 sides. To determine the shape of the African swine fever virus, you will first need to isolate a virion or sample of the virus. To do this, you'll have to perform a cell culture of infected host cells or inoculated embryonic egg cells. The cells, which are the growth medium, will then be harvested and the virions isolated by centrifugation. The virions or inoculated cells will further be analyzed by electron microscopy to confirm the virus's morphology.
The eclipse phase of the virus is the time interval between viral penetration and the production of progeny virions. It consists of the lag time needed to dissolve all protein components and take over the host DNA transcription mechanisms to create viral DNA before new progeny can be made. This phase is specific to this virus due to its dense virion core and its complex outer envelope. Essentially, the virus relies on specific internal conditions of the host membrane-bound compartments in order to break down its specific and complicated capsid. Once decapsidated, the virus can now reproduce in the cell, ending the eclipse phase. Variations and mutations. The complexity of African swine fever has been clearly demonstrated in the presence of 22 genotypes. As a result of the large genomic variation in African swine fever, mutations can occur. Often these mutations have led to new variations of the virus in different regions around the world. The type of mutations that occur are mainly the result of a gain or loss of genes belonging to one of 5 multi-gene families. The genomic variations among strands create various degrees of virulence and lethality.
Virus transmission. Warthogs are the natural host but cannot transmit the virus directly to pigs. Agar-agar ticks are the natural vector, and they can maintain and transmit the virus for several years. Virus transmission occurs through direct and indirect contact. So direct contact is through bodily fluids, which are oral, nasal, fecal matter, as well as blood, and indirect contact through contamination of farm equipment and clothing. Consumption of infected meat that is undercooked also allows for virus transmission. The virus can be airborne for up to 2 meters. This allows for transmission within a herd rather than between herds. Stable flies are also known to carry the virus. Viability of the virus for up to 48 hours within the fly. This also allows for transmission within the herd rather than between herds. Immune system evasion. So, the virus replicates and interacts with the host cell in a complex way. the virus encodes novel genes that allow for modulation of the host immune system, mainly through the production of specific proteins. Molecular mechanisms are used to evade host immune response. Some are: firstly, the alteration of interferon production. Interferons are released by the infected cells to alert neighboring cells to heighten their antiviral defenses. Secondly, inhibition of specific protein complexes, like the NF-κB, which is a critical protein complex in cell survival and preservation. Thirdly, through inhibition of nuclear activating factor in T cells. And lastly, through the modulation of host cell defenses. This is our bibliography, our online sources. Thank you for listening.